right, then thank you everyone for joining today's Matter to Life lecture series, a lecture series where we introduce all our fellows of the Max Planck School Matter to Life, and they are actually distributed on 14 different locations throughout Germany, which you can here nicely see on the map. So while I'm currently located at the central office in Heidelberg, I'm very happy to have Andrea Musacchio today with me, who's located at the MPI in Dortmund. And in order to get to know our fellows a little better, we have prepared, as usual, 2 questions to get to know him a little better before we then dive right into his research. So with this, Andrea, I'm very happy to have you here. And my first question to you is, can you tell us about your current role at the Max Planck School Matter to Life and how your work contributes to the broader Matter to Life field?
Okay, so thank you, first of all, for the invitation. It's a pleasure to be here. I mean, I would say sort of, okay, yeah, so in the entry level answer is that I'm currently hosting a student from the school, Adel Yusupov. And Adel is working on, in fact, on something that I'll discuss in the beginning of my lecture, which has to do basically with phase separation, which is a topic that is dear, obviously, to the school. I've been, in a sense, the public enemy #1 of this idea. I'll explain briefly why. But I have at least, I think, contributed to this discussion that is highly relevant, actually, also in the school. And I think that our work, contributes to the school because our biochemical reconstitutions show, in a sense, how complex it is to develop some sort of systems that mimic what happens in our cells. I mean, the complexity of these that will also emerge from the lecture. At the same time, they pose the fundamental question, how do you turn something that is inanimate into something that works at thermodynamic equilibrium into something that is alive, at least in the simplest possible terms or perspective, which is dissipative, that uses energy to do interesting things. So I think that the themes that we work on are very much part of the school.
All right, thank you very much. And then I have a second question. So what are some of the most significant challenges you have encountered in your career so far, and how did you address them?
Oh, God.
It was a little tricky one.
Okay, sort of, there's been loads of challenges from when I decided to stop studying literature and philosophy to study biology, and then when I turned into something sort of closer to a physicist in a sense, and sort of studying protein crystallography as a PhD student at the MBL. I was coming from biology, so I had to, and it was a time when you really had to jump into a new field, to be a crystallographer. So I think in general, the challenges have been pleasant scientific challenges, how to get into new field, how to incorporate new techniques into your lab, how to make sense of the complexity of biological systems. And I had, the usual complications in publishing and, making sure that, that people around me are happy, and at the same time, they're motivated to work and discover important things. I mean, I had, sort of for instance, the phase separation sort of discussions have been challenging in a sense that put me, sort of in the spot as a contrarian. I always try to speak my mind. I mean, sort of sometimes in ways that have annoyed people because I'm quite direct. Not so much. I mean, I'm quite direct when I know that people can defend themselves. So I try to be usually highly educated, but in general, when I feel that people should not fear that I criticize, I do criticize. And that's actually sometimes not taken very well, but I'll still do it. Yes. Okay. So there were challenges along the way, but by and large, I would say that I was lucky, very lucky. Yes.
All right, thank you very much for these great insights. And now we're very much looking forward for some research insights from your side. And with this, I'll hand over to you, and the stage is yours.
Thank you so much. So it's really a great pleasure to tell you more about our work. So let me Let me tell you, essentially, we work on cell division. I mean, and we do so using humans or human cells as model system. So there's beautiful lattice light sheet images of chromosomes as a cell divide. This must be a HeLa cell, so a tumor cell. But as you see, I mean, sort of chromosomes are divided very properly in these cells. Now, I want to simplify a little bit because there's a lot of complexity, and I'll introduce some of that complexity as we move on. But I will say that as cells enter mitosis, a human cell will, first of all, disassemble the nuclear envelope, I mean, sort of a membrane that essentially keeps the chromosome separate from the rest of the cell. And the chromosomes will condense and will try to achieve the configuration that is called biorientation that is shown here. Essentially, all the sister chromatids, so replicated chromosomes, are still cohesed, still attached to each other like Siamese sisters in the region of the chromosome called the centromere. And they biorient, which means they essentially establish connections to opposite poles of the structure of microtubules called the mitotic spindle, so the gray fibers here. So as cells get into mitosis, this process is incomplete. And in fact, it needs to start. And within 20 minutes, cells go from this prometaphase condition in which all chromosomes are sort of scattered around the cell to the condition that we call metaphase. Now, there's some features at these stages of mitosis that are very clear. In the early phases, there's no tension between the sister chromatids. I told you that they're cohesed, they're held together. So there's no tension. You can measure the distance between them, and it's the lowest possible distance you can measure. As you go to biorientation, so to metaphase, this distance is instead increased, and we say that the kinetochores are under tension, and the kinetochores are the points of attachment to the microtubules. During these attempts to biorient the chromosomes, the spindle checkpoint is active, and this is a pathway that originates at the structures, the kinetochores, to keep that cell in mitosis, essentially saying we're not ready to go out of mitosis yet. Whereas when we get into metaphase, so the alignment of chromosomes, essentially we have stable bioriented attachments and the checkpoint is satisfied, which means it's inactive. Now, what is quite extraordinary about this is that within the 20 minutes that a mitotic cell sort of uses to complete the process in humans, there is a dramatic change in the composition of proteins that reach these structures that I told you about, the kinetochores, that ultimately bind to the microtubules. So early on, we find a set of proteins, these and many others that are not indicated here. And later on, some of these, or the majority of these, are in fact replaced. I mean, they disappear or their levels are highly reduced and new proteins appear. And also the fundamental question, this is exemplified by these beautiful images that were taken, movies that were taken by Ted Salmon and colleagues a long time ago, where a protein, fluorescent protein, was injected into these cells, in particular this protein called MAT2, and you see localized at the centromeres, at the kinetochores very early on, and then basically becoming dissipated before the loss of sister chromatic cohesion that marks the end of mitosis. Okay, you'll see the sisters now finally separate. So long before, this protein has already gone, and this protein participates in this pathway called the spindle checkpoint. New proteins will appear, and so the fundamental question that my lab is trying to answer is, how are these proteins recruited? How are the binding sites generated in a way that is context dependent, so depends on the presence or absence of the microtubule? And I'll try to guide you to some aspects of this process. So just to summarize what I told you so far, the kinetochores are these structures that bind to microtubules, okay? They try to achieve biorientation of the sisters. So the two sister kinetochores on each of the sister chromatids is only bound to the proximal pole, but not the distal one. These are microtubules that connect. I mean, it's a little bit of a simplification. And when the process is ready for all chromosomes, you undergo sister chromatid separation. So the spindle checkpoint prevents essentially a situation like this, in which an unattached kinetochore is still present, and cystochromatic cohesion loss of cystochromatic cohesion has ensued. This should not happen because otherwise you lose this chromosome, it doesn't know where to go. The kinetochores try to correct essentially incorrect configurations of the attachments to microtubules. These are called syntelic or merotelic attachments. So there's a system of error correction that I'll mention briefly later on. There's the ability of the kinetochores to capture forces that are generated at depolymerizing microtubules and that propel at anaphase after the loss of cystochromatic cohesion, the chromosomes towards opposite poles of the dividing cell. And then there's a phenomenon that I will not discuss in detail, but on which we work very extensively, that is called epigenetic centromere inheritance. And the centromere is essentially the specialized locus on the chromosome where the kinetochore is assembled. And so I want to spend a few slides explaining what is at the centromere and how is the kinetochore built on this structure. Now, sort of for those who like sort of condensates, you should say sort of the centromere looks pretty much like a condensate. That is, it looks like a dot where molecules are recruited and concentrated. And so the centromeres are the specialized loci that promote the assembly of the kinetochore. You see them in green. And if you look, I mean, sort of you can see the chromosomes in sort of a little bit of staining. And these are the spindle poles. You don't see the microtubules here. So question is, what gives the centromeres their identity? The ability to maintain this identity, I will not discuss that in great detail, through cell division, and the ability to form a kinetochore. Now, I should say that, so technically the centromere can be regarded as a membraneless organelle. I mean, you agree that it looks pretty much like many other membraneless organelles that exist in the cell. So we can call them membraneless organelles, subcellular compartments, biomolecular condensates, and pretty much, I mean, sort of the structures that have been discussed from the nucleoli to all the structures shown here, many others that have appeared in recent years, are also membraneless organelles, subcellular compartments, biomolecular condensates. Now, when defining the centromere as such, the question that we want to ask is, well, what is required to assemble these structures, okay? Now, centromeres have all the features that have been attributed to biomolecular condensates, according to a very fortunate review by the Hyman and Rosen's lab in 2017 that defined biomolecular condensates. So they're mesoscale, so they're much larger than the individual components that make them. They fit the definition because essentially the definition was very simple. They concentrate biomolecules. So there's plenty of biomolecules that become concentrated to the centromeres. However, I want to say this clearly, there's absolutely no evidence that liquid-liquid phase separation is implicated at any level in the assembly of these structures. So we published in 2022 a commentary in Embryo Journal where we summed up essentially arguments to say that specific interactions and not liquid-liquid phase separation is the more likely driver of compartment assembly or condensate assembly. And in 2024, last year, a student in my lab started working, in fact, I mean, we published this paper that sort of meant to provide sort of the limits of, or demonstrate the limits of this idea. And as I said, my opinion in essence is that the solute-solvent interactions that are emphasized by liquid-liquid phase separations idea are unlikely to explain the assembly of biomolecular condensates. And one set of experiments that we did at this time, I'll show you in a second, has to do with demonstrating how, sort of why you need specificity in the assembly of these structures. Now, sort of when you define membraneless organelles, subcellular compartments, biomolecular condensates, I don't mind so much what name you use. Okay, this is really semantics. I mean, the name does not question the existence of any of these structures that were known to exist long before the liquid-liquid phase separation field providing an answer to how they assembled. So there's no question that they exist. We're not discussing these, and how you call them is not so important. What is important instead is what kind of theory you develop for how these structures form. So the liquid-liquid phase separation, sort of with the, say, if you wish, simplifying the oil-water paradigm, says that non-specific interactions are at the base of assembling the structures. Others, like ourselves, we say specific interactions, including multivalent interactions, are important for this. And here is not semantics anymore. It really does make a difference if you interpret the assembly of the structures one model or the other. And so we wanted to show these with the centromere in particular, because there's been a claim that a particular complex that assembles at the centromere phase separates there. And this was based on experiments in vitro, where indeed, I mean, we reproduced the original phase separation behavior of this object, but we wanted to demonstrate that this is really driven by non-specific interactions, by putting a non-specific competitor into the picture as a disturbing factor. The point being that, if it is this phenomenon here is driven by specific interactions, it should resist non-specific competitor. But if it is driven by non-specific interactions, it will not. And in fact, so what we did, which is not a very clean experiment, but so we did it in different ways, is to use an E. coli lysate. I mean, sort of at different concentrations. Consider that E. coli cytosol is at about 250, 300 milligrams per milliliter. And we observed that already at 15 milligrams milliliter of the lysate in these experiments, so 1/20th of the cellular concentration, we completely dissolved the phase separation behavior. Now, we repeated these experiments also with mammalian lysates, and we saw exactly the same. So without the lysate, you see these droplets, they disappear when you add the lysate. We added fetal bovine serum that has never really seen these proteins, like these are human proteins. I mean, an E. coli lysate or fetal bovine serum, they haven't even seen it. And even in this case, we saw dissolution of this phenomenon, okay? So we think essentially that these phenomena, for the most part, when analyzed in vitro with single proteins or a small combination of proteins, are artifacts from non-specific interactions. Now we show the generality of this by taking a number of other proteins that do phase separate in vitro. So these are real phase separation phenomena. In all cases, we could dissolve the phase separation with the lysates. It's very easy to demonstrate the generality of this. Okay, so given these, I mean, sort of I have now to provide sort of an alternative explanation for how these structures work. And I think when you ask the question, how is the centromere established, everyone will give you more or less the same answer, which is that you need to have this protein called centromeric protein A that was discovered by Bill Arndt and Naomi Rothfield a long time ago, and later shown to be a histone H3 variant, so similar to proteins that bind to chromatin. So this protein is there and is recognized as a landmark of the centromere. And many years after these initial discoveries, I mean, some people, including Don Cleveland's lab, but also other labs, looked at proteins that may physically associate with the SEMPE nucleosome. So they put a tag on SEMPE, pull it down, and looked at by mass spectrometry what proteins come down with SEMPE. And they found plenty of proteins. Many others were found. I mean, sort of this is from one lab, and other labs found more. But by and large, I mean, sort of the one thing that was always there was basically this set of green proteins, also called now centromeric protein letter. So some C, some L, N. I mean, you don't have to remember all of these. I mean, think of the green subunits until the appropriate time. I mean, but I won't go into a lot of detail here, but it's just to tell you that SEMPE is there, and then new proteins come and bind to SEMPE to be recruited. I mean, so this is binding, okay? Now, this sort of and other studies led to the idea that the kinetochore is a layered structure with the SEMPE variant at the bottom of it. This is in pseudo coloring, and it's the coloring is different from the one I use here. But imagine essentially SEMPE being roughly here, and here being sort of the green subunits. And then the yellow subunits here, the colors are the same, and I use also the same color for the microtubules. These yellow subunits, and so I should have told you actually, the green subunits are now known as the constitutive centromere-associated network, or CCAN. The yellow subunits that I haven't shown you in detail are 10 subunits in humans, but also in yeast, and they're called the KMN network. I'll show you in a second why. And within these yellow subunits, there's microtubule receptors. So the microtubules that terminate at essentially a metaphase end on this interface are essentially binding to the KMN network. So how can we make inroads on the organization of the structures, of these complicated structures? And many years ago, we decided that the way to do this would be by biochemical reconstitution. And so we thought, okay, sort of let's start identifying building blocks, sort of the bricks that would allow us to build the whole structure from sort of the individual subunits. And this took us a long time because we had to figure out, what binds to what, and how to make a stable subcomplex. I mean, this took a long time, many papers, many installments. And eventually we had a collection of stable subcomplexes that could be used to venture into reconstituting the whole structure. Once again, human kinetochore here. There's many more that are not shown here that we have in the lab and we handle in the lab, but this is already a good sample. And with this in mind, we thought maybe we can do not really a reconstitution of the full kinetochore, but a reconstitution of a module of kinetochore assembly. So the whole kinetochore, I already showed you this image. It's a very large structure because, think of a microtubule is about 26 nanometers in diameter. So this object is indeed mesoscale, okay? So how do you build such an object that is embedded in a complicated chromatin of the centromere region, which is made of, sort of there's between here and where the other sister chromatid will be, there will be another kinetochore sort of pointing downward. There will be roughly between the two sister kinetochores without tension, there will be 1 micron, so a lot of space, okay? So a lot of material in between. So how do you extract the structure from, what do you reconstitute really? And so we thought that maybe what we can reconstitute is a single module that could be convolved at multiple sites on the chromosome. And that presumably would contain basically a Sempei nucleosome and the proteins that I showed you bind to it, the CCAN and the KMN. And there was a reason to think that maybe a module like this exists because in Saccharomyces cerevisiae, the kinetochore is exactly this. Essentially with a similar composition to the human kinetochore, so roughly the same proteins. I mean, basically, this is what, it binds to single microtubule. And so we thought maybe we can do this with the human proteins as well. And it turns out that this is indeed the case. And this is a gel that was taken from this paper from Kai Weilstein, who was a student and then a postdoc in my lab, where we have reconstituted essentially from the building blocks that I showed you before, a structure that looks like, could be possibly a module like this. So what do we have here in this structure? Just to simplify, we have the Sempei nucleosome, we have the green subunits, probably 2 assemblies, because the nucleosome is twofold symmetric. And then through tricks that I will not explain and that have to do with phosphorylation sites, etc., there's an increase in the stoichiometry of the microtubule binders, which, the NDC8 is really the complex that contains the microtubule binding site, and that we think is present in 6 to 8 copies per Sempei nucleosome. So it's a small number of molecules, but this multiplicity is absolutely essential for proper microtubule binding. So what do you do with a reconstitution like this? Well, you can do, high-end biochemistry. You can use it to, identify binding factors, phosphorylation sites. You can do a lot of stuff. You can do structural biology. So you can try to use it to look at the structure of the kinetochore. And then in the long run, you may try to turn it into an alive structure, if you wish. That is, you turn it dissipative in such a way that it could read microtubule binding and give the appropriate response, create and dissolve the binding sites that I told you about. I mean, that's our goal in the long run. So structural biology, we did a lot with this. I mean, just 3 years ago, we published a structure that we now have also refined to include DNA of the CCAN, so the green subunits. And this came a little bit as a surprise because we thought that CCAN was a nucleosome binding moiety. In reality, it's a DNA binding moiety that binds to a nucleosome indirectly through one of the subunits, SAMC, and probably the nucleosome is displaced by some distance. So there is a connection, but not a direct connection as we had imagined. Okay, so this connection is created by a protein called SAMC. Now, this is a detail, but essentially CCAN is a DNA, binds to naked DNA through the structure. There's, we call this the vault. It surrounds essentially the DNA. And then we also solved structures over the years of the outer kinetochore, the yellow subunits that came a network. And again, I will not go into detail for the interest of time. I'll just tell you that, a long time ago already, we had created what we call the NDC80 bonsai, which was essentially a dimer of what is normally a tetramer, where we could use it together with Evan Nogales a few years later, not only to have a structure of a simplified form of the NDC80 complex, the microtubule binder, but also one that we could use to decorate microtubules in this sort of crystallography, hybrid crystallography EM effort, early on effort, in fact, and we gain an understanding of how this complex binds to the microtubules. So we solved other parts of these. I just show you this, what you see here now is a cryo-EM map that will be fitted essentially with elements that we had solved before, and also AlphaFold 2 models to just give you a hint of what the structure of this module here may look like, which is essentially a submodule of the kinetochore. And so you will see now the models being built and fitted into this density. Here is where you create a connection with the inner kinetochore at this end. And now you will see actually the NDC80 complex is this wild beast that binds microtubules at this end. So these guys are really antennas that bind microtubules here and connect to the kinetochore at the other end. Okay, so this gives us now a composite of the organization of a kinetochore module. What I'm showing you here is not the whole structure, which is even more complex, and structural work will be continued to ideally reconstitute and image this structure shown here with all the modules being present, all the parts, if you wish, being present. But it gives you an impression. And there's a large intrinsically disordered region within the kinetochore on a protein called KNL1, which is a binding platform for the signaling proteins that I told you about, or at least a few of them. Okay, so how will you build then this mesoscale structure? This biomolecular condensate is presumably by convolving the structure that I told you about at every SAMPA nucleosome that you have in your chromatin. So, these are two modules, and the real structure has many of them. So you can this is one example of how, you can build a mesoscale object from a nanoscale module, but there's many other ways in which you can do this. I mean, you don't need phase separation, okay? So you really don't. Okay, so now the outer layer, so that Cayman network, the yellow proteins, recruit all the regulators that I told you about. I mean, the ones that signal early on and the ones that are present when the microtubules have arrived on the kinetochore. So the binding sites are created and regulated on the outer kinetochore, which makes sense because essentially the cell is trying to monitor microtubule binding to the kinetochore, okay? So now, so it's a complicated slide, but it shows you what you've seen already. All the subunits bind to the KMN network, to the structure here. And if you eliminate, this and that, and you do an experimental localization, these are the kinetochores, these are fixed cells. You see the kinetochores here. These are two of the proteins in these groups here. And if you eliminate the KMN, there's none of them. And this is true for all of these proteins. So they're all recruited there. So if you want to make sense of these on molecular terms, understanding what happens, you need to know exactly where these regulators are recruited and how the recruitment is regulated, okay? Because you need to be able to do separation of function mutants. You'd like to essentially eliminate one regulator at a time. You cannot get rid of all of them at the same time. So a fundamental concept is that the pathways, these regulatory pathways, probably depend on the same sensory apparatus, and that the apex of this system is a protein kinase, so an enzyme that uses ATP to transfer phosphate onto target proteins, okay, called Aurora B kinase. And it's been shown already a few years ago that Aurora B can form a phosphorylation gradient within the depth of the kinetochore that goes from the centromere to the outer kinetochore in the opposite direction. So there's more phosphorylation towards the centromere than there is in the outer kinetochore. But especially the presence of a microtubule will subtract substrates from this gradient of Aurora B in such a way that stuff that is closer to the origin of the gradient will continue to be phosphorylated, but stuff that is on the outside will stop being phosphorylated also through the action of phosphatases. Essentially, under these conditions, the phosphatases are repressed enough that they cannot remove those sites. But when you move, just by sort of traction, if you wish, of pulling of the kinetochore outside of the gradient, sort of along the gradient, now the phosphatases can reach sites that were not reachable under these conditions, okay, to dephosphorylate. And the kinase has more and more problems phosphorylating these. And this is known to stabilize kinetochore-microtubule attachment. Now, I will not try to clarify how this gradient is created and what is the evidence. We don't know exactly how it is created, but there's a lot of evidence in favor of this idea. So now take it for granted that Aurora kinase works upstream, and just downstream is another protein kinase called MPS1. And so MPS1 is a protein kinase, again, like Aurora, but with a different specificity that is completely dependent on Aurora B for its localization to the centromere. And this protein is clearly required both for bioorientation and for the spindle assembly checkpoint, okay? The recruitment mechanism is debated and will be essentially my last slides on this topic. So first of all, let me show you that it is true that you require Aurora B kinase to recruit MPS1 to the kinetochore. So here in red, you see MPS1 localizing to the kinetochore. This is a marker of the kinetochore, so you know where your kinetochores are. And you see MPS1 being localized there. This is your control. Now, you can inhibit MPS1 activity, and MPS1 continues to be recruited, so it doesn't need its own activity to be recruited there. But if you inhibit Aurora kinase activity, now MPS1 has disappeared, okay? And, okay, so whether or not you inhibit MPS1 activity. So Aurora B is required for the recruitment. Now, as I said, this protein works both in the checkpoint and in bioorientation. I didn't tell you much about the molecular biology of the checkpoint, which is very interesting. And the molecular biology of bioorientation is even more interesting, but even more complicated, just to keep it simple. But I'll explain how we can reach this conclusion, okay? And to do this, I mean, sort of first of all, to check the involvement of MPS1 in the checkpoint, you can monitor the degradation of cyclin B. And cyclin B is essentially the engine of mitosis. So if you want to go out of mitosis, you need to turn it off, and you turn it off by proteolysis. So that if you have a sensor in the cell that tells you how much cyclin B you have through fusion to a GFP, okay, you will see that the levels are high until cells have achieved biorientation, when they start degrading cyclin B, and cyclin B very rapidly, within minutes, is destroyed and the cell can leave mitosis. Now, if you want to see what happens when you activate the checkpoint, you give cells a spindle poison, so a poison of microtubules that prevent essentially the spindle from working, and therefore you never degrade cyclin B because the checkpoint is active. And the checkpoint wants to maintain cyclin B to maintain cells in mitosis. And you see this. There's very slow degradation of cyclin B, but there's plenty of cyclin B in the cell to maintain it in mitosis. Okay, so the checkpoint is active. And here, this is my student, Stefano Santaguida, a long time ago, when he was a student in my lab. He discovered an MPS1 inhibitor called revarsin that everyone uses now to inhibit the checkpoint. And in this paper by the Pines lab, they use revarsin to inhibit MPS1, showing that when you do not have the checkpoint, cells leave mitosis earlier than cells that complete mitosis normally, okay? So you're sending the cell into precocious mitotic exit when the process of biorientation is not completed yet. Okay, so MPS1 controls the checkpoint. Now imagine that you have a way to keep cells in mitosis when you inhibit MPS1. You can ask under these conditions whether MPS1 also controls the biorientation of the chromosomes. And this is exactly the experiment. Here we inhibit mitotic exit by preventing the degradation of cyclin B. But we add revarsin to kill the checkpoint and to kill MPS1 activity, okay? But the cell remains in mitosis. And now we go and look at whether chromosomes are properly aligned, forming a biorientation, an equatorial plane or not. This is your control in this assay, so all chromosomes align at the metaphase plate. When you inhibit MPS1, many chromosomes cannot align to the metaphase plate, which means that MPS1 is required for the checkpoint, and it is required for biorientation. So it's the same sensory apparatus. So where in the kinetochore does MPS1 bind? And if we knew this, we could reveal an important regulatory role of MPS1. So we could tell essentially, well, presumably, you know, this binding site that is so important for the regulation of this process is being there is probably regulated this way. And so there are two prominent studies 10 years ago suggesting that MPS1 binds at the microtubule binding site of the Kamen network on NDC80. And by binding there is subject to competition by a microtubule. So a microtubule that binds here will occupy the site that MPS1 wants to bind to, and therefore MPS1 will fall off the kinetochore when the microtubule is bound. So we debunked this. It's not true. And we did this with an approach that we use all the time. And this is another example of how you can use recombinant proteins. We use them through an electroporation experiments of the recombinant complex with a chlorophyll into cells that have been cleared of the endogenous complex. So the experiment is here, you know, is an RNAi in this case against the NDC80 complex. NDC80 is there when in control conditions, MPS1 is there. You remove NDC80, there is very little MPS1. Now you restore NDC80 with the recombinant protein, okay? Recombinant proteins, 4 subunits, electroporated. The complex goes to the kinetochore and recruits endogenous MPS1. And now you do an experiment with a mutant in the region that binds to the microtubule, and that you would expect, if competition is important, to be also important for MPS1 recruitment. But sure enough, MPS1 is still there. So it wasn't true. So we debunked those papers, but now we were left essentially with the responsibility of showing where does this protein bind. And it turns out it's a very complicated process. There's many binding sites, but to show you what happens when you have identified one of these, is that it is a site in this tetramerization domain. Now, when we do the same experiments I just showed you, NDC80 mutant is at the kinetochore, but MPS I, contrary to what you see in the control, MPS I cannot be recruited. So we have identified now at least a couple of sites where MPS I binds. It's a very complicated process because Sabrina, who's a PhD student in the lab, has shown essentially that MPS I binds to at least 3 different sites on the combination of the NDC80 and canon1. And so this is, I think, my final slide. The question we want to ask, and Jason also PhD student in my lab, he tries to apply forces on NDC80, which are essentially in an optical tweezer, which would be the forces that normally microtubules apply onto the kinetochore subunits that are bound to the microtubules. So the entry point of the force within the kinetochore is essentially the NDC80 complex. And so we asked in an optical tweezer where you can apply forces on single molecules, what happens to the NDC80 when you put it under force? And this is still unpublished, but we have some very interesting results because there is some force-dependent behavior of this complex, although we still do not fully understand what it means. But the fundamental question that will follow up is how these structural changes on the NDC80 coupled to the gradient of phosphorylation that I told you about regulate the phenomena that I discussed. And so sort of this is the summary slide. So bioorientation and the checkpoint depend most likely on the same sensory apparatus. Aurora B is at the apex together with the main mitotic kinase, CDK1. I did not explain that. But the holy grail in kinetochore biology is to understand how the sensory apparatus work. And we hope to be able to reproduce it really in vitro. And so biochemical reconstitution of the kinetochore may help us make a fundamental inroads eventually into the problem of how the cell senses bioorientation. So I acknowledge the work of the people who did the work I presented today. So I'll go quickly here to this slide with funding sources, and I wait for questions if you have them. Thank you.
Right. Thank you so much for the presentation and the insights in the current ongoing research. Now the stage is up for questions. So I'm asking everyone in the audience to post your questions in the question and answer bubble on the bottom of Zoom, and I will read them out to Andrea, and he will gladly answer them.
Sorry that this was a bit long, I guess, and a bit longer than usual, or I ran over time. Probably everyone does. I console myself. Okay, it's a complicated problem. Should I stop sharing, by the way, or
sorry?
Should I
You
stop?
can if you like to. If you think that some of the slides might help.
Maybe that some of the slides, okay, yes, they could be useful.
Right, that's why you can also keep them. There's a question in the chat now, so I would read this out to you. I apologize if I missed this in the beginning, but are there particular diseases where the sensing mechanism are known to be relevant?
Yes, well, there's especially cancer, because I mean, okay, so that is, it's hard to say that is genetically, the machinery that I told you about seems to be protected, even in cancer cells. I mean, there are mutations in some of the proteins, but I would say it's nowhere close to the frequency of mutations in known oncogenes or tumor suppressors, okay? Like, RAS, P53 or PI3 kinase, nowhere close. And we think that the reason for this is that cancer uses chromosome instability, so essentially a high rate of chromosome loss or gain, to explore new variants, yet it cannot afford getting rid of the system completely, because in fact it could be over-reliant on these pathways, because otherwise the genomic instability that will ensue would be so great to make even cancer cells inviable. And so usually the pathways that I told you about are conserved or are kept functional in cancer cells. Maybe they're modulated to work a little bit less, but by and large, you know, they work well. Now, in cancer cells, however, this rampant chromosome instability is very high. I mean, sort of, you know, if you look at the genomes of cancer cells, they acquire chromosomes, lose chromosomes. I mean, there's a mess in their chromosome complement. Exactly how you get there is not terribly clear, but one has to remember that there's thousands of proteins that work in mitosis and that could be affected in ways that generate this instability. So it's extremely difficult to, you know, identify the causes of chromosome instability in cancer. But certainly, if you take, you know, sort of feature of cancer, genomic features of cancer, chromosome instability is by far the highest. And sort of you always have chromosome instability in cancer, almost always, especially in aggressive cancers. So the short answer is, I mean, the pathways, the regulators, the regulatory pathways seem to remain functional, even in cancer cells. But cancer cells are highly mutagenic from the perspective of losing or gaining chromosomes.
And while we were speaking, there are a couple of more questions were coming in. So the next one is, what is your current theory on the sensory detection mechanism?
Okay, great question. Well, I mean, sort of, okay, maybe if I can go, let me see if I, oopsie, no. Oh. I thought you'd be asking questions about phase separation, but instead, it's nicer that you ask these questions. Yeah, so the sensory apparatus somehow has to do with this Aurora B gradient. And you know, the way that this is established is we think is that basically the complex that holds onto Aurora B is made of a localization module and a kinase with a long intrinsically disordered region in between. And the localization module goes here essentially at the beginning of the gradient and sets basically where the gradient, where the highest activity of Aurora B will be. And then sort of we call this the dog leash model, Aurora B being attached to the end of this INSEMP, intrinsically disordered protein. The farther out you go, the less space it can explore per unit of time. I mean, it's a very simplified idea, but imagine a sphere that can be explored by Aurora B. The farther you go from the center, the lower the probability that Aurora B will be found there. Okay, so this is how we think you establish a gradient. So we don't think that there is a diffusible gradient. It'd be impossible, I think, to build a diffusible gradient over 100 nanometers. It's too small. You would need fantastic rates of activity. So the gradient is probably generated by, you know, localizing something there. Now, these, of course, I mean, sort of as you said, I mean, to generate a gradient that changes over time, you also need the phosphatases. And it could be essentially that, you know, imagine you're a site here. Okay, if you're a site here, Aurora B keeps phosphorylating all the time, and the phosphatases do not have the ability to completely dephosphorylate a site here. But a site distant from the center of the gradient that will be moved farther out will be explored by the kinase less frequently, and now the phosphatases have the ability to remove the phosphates. These phosphates may act either as binding sites for something or as modulators of the strength of kinetochore-microtubule attachments, so that when the microtubule pulls, essentially, you know, so the kinetochore knows that it needs to attach to the microtubule more strongly. And the second hypothesis we make is that the yellow subunits change their conformation and their force to accept new binding partners that could not bind to the protein when it's not under force. So these two theories together might explain essentially how the sensory apparatus works.
And there's another question to the phosphorylation gradient. Not sure if that's also partially now answered already, but so the person asking is not completely sure about the phosphorylation gradient works. So how is the aurora kinase activity depending on distance from the centromere, and what determines if the level of phosphorylation is big enough that the endpoint of the complex will be phosphorylated?
Yeah, so those are all very good questions because we start from the assumption that Aurora B does it, okay? And we think the reason is that the localization module that I told you about with Aurora B being attached, we call it dog leash. Imagine a dog, okay? And imagine that this is the owner. So the dog, can walk a certain distance, but at some point the leash, limits the distance that it can go. And obviously, if you think of the function as a sphere, the more volume you have, the less likely you are to explore it. I mean, this would be the hunch. Now, the question is, how do you localize the owner of the leash and the dog at the right place? And here we know roughly how you do this because we know that there are essentially some localization cues that are located at the centromere, okay, where the localization module can go. So with a protein, so consider that this is 120 nanometers, what I told you about. I mean, the green subunits and the yellow subunits. I mean, the yellow subunits, so the antenna is some 85 nanometers, and then you have, some 25 nanometers of the globular guy at the bottom, some linkers. I mean, you have, roughly these distances, and you have this very long intrinsically disordered region of incent that may allow the dog, to go inside the structure in a gradient dependent manner.
Next question. In the analogy that you did about hardware and software, hardware will be the biology structure and the software, the specific proteins that modify the biology structures.
Yes, exactly. So I didn't even read the title, but essentially was exactly the idea. That is, this is the hardware and this is the software. And the hardware is designed to find microtubules, and the software is designed essentially to making it do it properly. And making this happen properly means that you don't bind too strongly, too early to the microtubule, because after all, maybe I should have. So I keep showing you a single module here on microtubule binding, but there's obviously two, right? Because I mean there are the two sisters, and the sisters are linked to each other, and they need to know. Okay, so let me go to the first slide here. These two guys need to know that they're properly oriented. That is, that they have achieved biorientation. So they need to know that they're under these conditions. And if they're in a wrong condition, and we know really there's a force sensor because Bruce Nicholas did these beautiful experiments a long time ago. We still don't know how the sensor works. We hypothesize, but essentially, if you have incorrect attachments, you need to be able to recognize them and correct them. So you don't want to bind to the microtubule too early, too strong. You want to make sure that you have a way to recognize when your sister is bound to a microtubule, or it's not bound. And this is, I mean, sort of the fundamental problem that the sisters had to solve to be able to biorient. So this guy here needs to have a way to tell whether the sister is bound or not to microtubules that are directed to the opposite spindle poles. And what is amazing about this is that what you have in between doesn't matter. And it's absolutely clear from a number of evidences. It's not what you have in between that matters, is what each kinetochore senses. I mean, this kinetochore has the ability, regardless of what is here, provided that at some point tension can be transmitted through the material, whatever material it is, at some point it needs to resist. This kinetochore needs to know that there's resistance from the opposite sisters. So independently on what is in between, this guy by itself can compute its state based on information it receives indirectly by the sister. It's quite extraordinary. And I think essentially if I have to provide an analogy, the difference will be between sort of lifting my hair with my fingers and lifting my entire body through my hair. That will be the difference. I mean, obviously the levels of force are very different. And so something equivalent may be happening here. That is relatively easy to pull within the kinetochore, but the level of resilience or resistance that will ensue when the other kinetochore is bound is much higher. And this is actually what stabilizes the attachment. So the regulation that I told you about is somehow sensitive to this.
Okay. Next question is, how are these mechanisms related to aging?
Yeah, so these mechanisms are related to aging somewhat indirectly, but any form of genomic instability and genetic instability is related to aging. So aging is a consequence of acquiring genomic errors of different kinds. So genomic errors during DNA replication, but also during chromosome segregation, so that a lot of the aneuploidy, so the changes in chromosomes. So we always make the assumption that our cells are all identical, but genomically, but our cells are not identical. And if you look in tissues that keep replicating in your life, I mean, you go to the lungs or to the intestine, and so on and so forth, and the blood, you'll see that there's a lot of differences between cells, because I mean, especially as you age, because you accumulate mistakes, right? So if you have a system, and in fact, I mean, this is well established. I mean, there are mutations in proteins that I told you about, and some of that I showed you. For instance, this BABR1 there are mutations that cause a quote unquote is a disease called is a progeric disease. I mean, sort of is a disease where people acquire traits of age earlier than, let's say, quote unquote, normal people. So it's a disease because it's a progeric disease. So it's an accelerated aging. And it has to do with chromosome losses or gains, very clearly.
Okay, so since we already have 4 o'clock, I would need to ask you how much time you still have, because there's still 4
I
questions.
have time.
And I'm fine to continue
I'll be
asking.
totally pleased to stay. If the students want to stay, I'm totally happy.
Then I'll just continue asking you. So the next question, the person was wondering whether the interaction of NDC80 to MT depends on MT properties, such as specific cross-translation modifications or conformations.
Yeah, that's a great question. We don't have a clear answer. So the end of the microtubule, because there's a handful of NDC80 complexes, right, that do these. I mean, there's probably, per module that binds microtubules, there's probably 8 of them. So what we think the NDC80 recognizes in reality is the bending end of a protofilament on the microtubule. So we think at the 3-4 NDC80 complexes that can sort of get staggered onto each other. So they sort of form a longitudinal stack. And that binds, we think, to the ends of the microtubules, which sort of peel out during polymerization and depolymerization. We're not completely sure about this because there's no, everything that has been done in terms of structural biology so far was by decorating the lattice of the microtubule, and NDC80 can bind the lattice. But from indirect considerations on the speed of microtubule depolymerization when these proteins are bound to the end of the microtubule and the microtubule depolymerizes, you can make the conclusion that probably NDC80 binds to the peeling protofilaments. Post-translational modifications, we're not certain about. I mean, sort of this has been a mystery, but I would imagine that it doesn't make a difference because this system is completely ubiquitous, whereas, the modifications depend a lot on organisms. So I would say that probably I wouldn't expect major differences, but you never know.
Okay. And the next question, if the KMN network is structurally rigid, stable, how is the reversal of kinetochore-MT attachment mediated? So especially if the early stages when error correction is active.
Yeah, so beautiful questions all along. I didn't explain this very clearly because, I mean, in the interest of time, I had to cut and I already run out of time. Let's see if we play this. Right here, you'll see soon a tail of the NDCD complex that is a target of phosphorylation of Aurora B, and we know that this does affect, okay, here, the tail. This has 5 or 6 phosphorylation sites by Aurora B. And we know that if you mutate those sites, this NDCD complex binds to the microtubules much more strongly. So what we know essentially is that this fragment, which partly binds the microtubule and probably allows the stacking of the NDCD complexes that I told you about, so it favors it by sort of connecting to the neighboring subunits. The phosphorylation sort of modulates this to prevent actually, a too strong binding from the beginning. I mean, sort of initially you want the binding to be strong, but not too strong, in such a way that if you mutate these phosphorylation sites, you get a lot of alignment errors. Essentially, the chromosomes cannot align properly. So this has to happen slowly. But we know, given that gradient idea is also based on the phosphorylation of this fragment here, because this is very heavily phosphorylated beginning of mitosis. But if you go at the end, when chromosomes are aligned, there's very little phosphorylation. So the gradient operates, one of the sites on which it operates is clearly this N-terminal tail of NDC80 to modulate the strength of binding to the microtubule. We still don't fully understand how this happens. Does this answer? I hope it answers the question.
We'll see. Maybe otherwise we'll get another one in the chat. Okay, then I would continue with the next question. So regarding the LLPS and condensates, I understand you're a critic as not targeting the existence and relevance of biomolecular condensates, or however you might call them, but focusing on what interactions give rise to these structures. If the condensates rather arise through direct specific interactions than through unspecific interactions, what implicates does this have? Or in other words, what in your view wrong conclusions are drawn from the pictures of LLPs as driving forces for condensate formation?
Right. Okay, this is also an excellent question. So yes, I mean, you could call them biomolecular condensate. I don't care how you call them, and that's why I say semantics. Provided you're not implying that because you call them biomolecular condensates, then they form by, LLPS. You have no evidence. It's very difficult to demonstrate this. Now, my critique is, in essence, that the entire field has forgotten about half of the story, which is, what is the solvent? I mean, if somebody comes up with a paper in Cell and says, we discovered the phase separation grammar of water, I would say you haven't discovered the phase separation of grammar of water, because there will be as many phase separation grammars of water as there are solvents for water when you dissolve it into something else. If you dissolve it in oil, you'll see phase separation, but if you dissolve it in alcohol, you won't see it. So the field decided that they could assume that there are phase separation drivers. I mean, I took these, from Cliff Brangwyn's. I mean, there's some of the top people in the field. Now, sort of they're claiming in this review that it was taken up essentially, essentially this is how the field thought about this. Essentially, they're saying that Phase has intrinsically disordered regions with residues that can somehow create an orthogonal compartment assembly from other, sort of proteins that also phase separate, in their opinion, using different interactions. Now, if you ask me, this is absurd. This will never happen. It's absolutely impossible because, I mean, these interactions are non-specific. So how do you verify whether your non-specific interactions can ever bring a protein X that you call the phase separation scaffold into a volume of space where the protein is phase separated? Okay, so you need to add the right solvent. And the right solvent is the cell, is the cytosol. Now, I don't like the cytosol experiment because you don't know what the lysate does to your protein. When you add the lysate, essentially, you could add proteases, nucleases, and you name it. I mean, it's not a nice experiment. But by diversifying the type of non-specific competitor, we can at least say, but look, I mean, sort of, it looks like whatever we put in there that is non-specific is not designed, to bind. It doesn't do anything. Okay, every time we do these, we lose phase separation. So how do you explain this? Now, obviously, the assumption is that under the same conditions and other interactions that is instead specific will survive. But I think there's every reason to think that that's the case because specific interactions is exactly what the cell uses to perform function. I mean, it's the stuff that I told you today. And we knew these long before the phase separation field developed. In other words, we did not need that theory to explain how proteins concentrate in the cell. We had, perfectly plausible ideas for how it could happen. So I think it was, just seeding a lot of confusion without ever coming to the point because, the reason why you get stuck is that, first of all, I mean, function is evolved in biology. And what it means is that, you need to be able to make mutations. And if you come up with something like this idea, intrinsically disordered things that interact with each other, an intrinsically disordered protein will never want to interact with itself. I mean, it's a crazy idea if you think about it, because there's no spatial coherence. They can go anywhere. If they have an isoelectric point that is different from 7, they will repulse each other. They'll rather go to an IDR with an isoelectric point in the other part of the, of the spectrum relative to pH 7, relative to neutrality. So why on earth would these proteins do this? It's just complete nonsense. And sort of it's an idea that, sort of was born out of, obviously, of not having ever seen a protein in your life. Because, I mean, you cannot be so naive about this problem, seriously. And I know I'm right. I mean, there's no doubt. I mean, so I'm sorry to be so arrogant, but I'm arrogant about this because I know proteins. It simply cannot work. And at this point, I think, we have pretty good evidence that that doesn't work. Right? So it simply doesn't. And I'm sure, do the test, on any protein that is a staple of phase separation, it will undergo the same fate. Not multivalent proteins that interact with each other through specific interactions. I mean, the test is very simple. If it is about phase separation, the solvent must be factored in. If it is not about phase separation, you can probably reasonably forget about solvent. It's as clear as I can't formulate it more clearly.
Okay, thank you very much. Then we have one very last question, and this is actually not on the presentation, but to the discussion we had before with the 2 questions on your career and the challenges you were facing. And the question is, so you earlier mentioned that you started out studying philosophy and then you transitioned to biology and physics. And the person is curious, what was the transition like for you?
The transition was, I was suffering because I came, I never thought I'd become a scientist. And so I was actually neglecting science as a high school student. It was a disaster. But then I started studying, topics in literature that were very technical, like paleography, linguistics, and I realized actually also the beauty of sort of becoming technical about things. And I told myself, you know, there's no way to solve any problem if you don't have some training in science. At that point, I was a little bit afraid because you know, my background was not really developed in terms. I thought maybe I could study physics, but I was really afraid that my math, you know, would take too long to develop, and so I started, you know, teaching myself a little bit of math, not much math, but enough, you know, to understand Fourier transforms or you know, geometric transformations. I mean, just to grasp the concept and being able, you know, to write an equation and understand more or less what's going on, sort of as a PhD student, which is actually, you know, so you have plenty of time to do. You know, obviously you have to work in the lab, but you also can spare time to do other stuff and learn new things, right? I mean, you learn to learn. That's actually what PhD students have to do. You learn to learn. You come out of university, you should be able essentially to take any text and dissect it. And right now you have more than text. I mean, we had text, complicated text. I mean, you have lots of videos and tutorials of all sorts to help you out. Yeah, so it was tough in the beginning, but I was determined to finish because I was getting old. So I told myself, you know, come on, give it a kick. So, but I have a lot of understanding for people who are curious about other topics because I mean, sort of, I bring it into my soul. And if you have an interest in other topics, you should always cultivate them because I think a novelty comes also from the ability to incorporate, you know, information from other sources at all levels.
These are really nice closing words. So thank you very much for your presentation.
My pleasure, and thanks for those who stayed until the end. It's been a pleasure.
Yeah, thank you very much also for the audience for all the great questions and the great discussion.
Yeah.
I think we now have a little summer break, but then we'll soon back with the lecture series. And yeah, check out our website for the next dates. Looking forward to have you soon in the next presentations. And Andrea, thank you very much.
My pleasure. Thank you. Thank you for hosting me.
Thank you so much. Have a nice afternoon, everyone. Bye.
Bye.